Welcome, everyone. Good afternoon, good morning, good evening, wherever you are in the world. Thank you for joining us for our webinar, Methods Sharing Platforms for Reproducible Research. My name is Emily Golden, and I'm the co-chair of the EBTC Open Science Working Group, and I'm joined today by Dr. Emma Ganley and Dr. Paul Whaley, our speakers. So for those of you who are not familiar with EBTC, I just wanted to give a brief introduction. EBTC is the Evidence-Based Toxicology Collaboration, which is an international collaboration that improves how we create, use, and publish toxicological and environmental health research. And EBTC is organized according to 4 themes, the first of which is raising research standards, so really just striving for that high quality research. The second is making sense of evidence, and this is the encouragement of the use of things like systematic reviews and evidence maps. And then the third theme is improving access to research, which really sort of encourages the use of fair data principles and open science. And then the fourth theme is advocating evidence-based decision-making, which is the policy piece of EBTC. And associated with each of these themes is a working group. They're listed across the bottom here. But today's webinar is brought to you by our Open Science Working Group, and you can join the group if you haven't already by scanning the QR code in the upper right-hand corner. And just a little secret, all 4 of the working group registrations are there, but we hope that you'll join our Open Science Working Group. And just a couple of more details about the Open Science Working Group itself. It was established to improve our understanding of how computers can make toxicological research work better. So I'm sure that many of you on the call can appreciate the fact that toxicology is sort of stuck in this paper-based paradigm, and we really want to get away from that to a computer-driven approach. And we think that this can start to be solved by planning and reporting of studies with computer-driven solutions. We think that those show a lot of promise and a lot of innovation. And so today's first speaker will discuss one of those computer solutions, which is Protocols.io, a platform for recording, sharing, and improving study methods. And we'll hear about that from Dr. Emma Ganley. And she is a longtime Open Science Working Group member and has extensive experience in the publishing field, where she has served in numerous editorial roles, including as executive editor of the Journal of Cell Biology, as well as the chief editor of PLOS Biology. She joined Protocols.io as the director of strategic initiatives in early 2020. And after her presentation, we'll hear a case study by Paul Whaley, who happens to be my wonderful co-chair for the Open Science Working Group. And he has been working with EBTC since 2017, and he also serves as editor-in-chief of Evidence-Based Toxicology, which is the official journal of EBTC. And then after his case study, he'll also touch on how to publish methods templates. And then, of course, we'll leave some time for questions from the audience. So with that, I will go ahead and hand it over to Emma.
Thank you. I will see if, let me see if I can pass the first test, which is sharing the right window. Okay, so I have got some slides, which you're hopefully now seeing the first one of, and then I will show the platform a little bit too, because I think it's better actually when you demo it live. So as long as my internet complies, that's the plan. So yeah, thanks for the intro. Thanks for letting me present to the group. It's been fun being involved. I don't always make the meetings, but I love that there's a big open science, open research push with these groups. So I will tell you a bit about Protocols.io, but also the importance of sharing methods. And I'll start there. So why is it that we care particularly about having methods shared in research? So these are tweets on the left, one from a biologist, and in the middle one from a physicist. So what we commonly see is that in published research papers, the methods details are terrible, lacking. They often just cite an earlier paper. So that's the instance in both of these, is where you look in a paper from 97 for that biology example, and it says as described in paper in 96, as described in paper in 87. Maybe the 87 paper is paywalled, maybe it's actually paper only, and some poor PDF version may be possible to find, but actually maybe not at all. So you may not actually get that information. Similar with that physicist devices as fabricated as previously described, and the jump back to multiple other papers to eventually find that the first original paper said that devices were fabricated with conventional methods. So I can't find, access, or replicate experimental or methodological details. There's a really nice study that the Center for Open Science and Science Exchange undertook a few years back, where they wanted to reproduce all of the experiments from about 50 published perceived high impact cancer biology papers. So they had all been published. The people undertaking the project looked in the papers, obviously, but also contacted the authors of those papers to see if they could get to the information of the experiments to try and replicate them. And there's a really nice write-up about this in The Atlantic, excerpted here. So the hardest part by far, figuring out exactly what the original labs actually did. Papers have method sections that should include recipes for doing same experiments, but often those recipes are incomplete, missing out important steps, details, or ingredients, and in some cases, they're not described at all. And not only were they not in the papers, but actually the researchers themselves didn't have the information. So they hadn't kept good records in the labs. They didn't actually know the specifics, all of the kind of tiny details of how those experiments were performed. So the long story short, they stopped this effort after they'd tried just 18 of those 50 papers replicating the experiments because it was too time and money expensive. And in a more recent write-up that's on the Center for Open Science's website, they actually note that from those 53 papers, 193 experiments, 0% of the protocols were completely described. So we do have an issue. We're not very good at getting the methods information out there, and we're actually maybe not very good at keeping a record in our own labs of what that information is. I always love showing this, not least because I like baking and eating cookies. Also with the virtual webinars, it's the way of sharing cookies that we would have had if we were all in person. This photo shows cookies that were all baked from the same recipe, but for which minor tweaks were made to the recipe. So if we're thinking scientifically, phenotypically, they're obviously all very different. Quite clearly, some look more appetizing, some look more burnt, some maybe not so tasty. Now, if cookies are the data and the results, the analysis of all those phenotypic changes, the size, the thickness, the texture, you can actually only interpret and understand why they've come out like that if you know the tweaks that were made to the recipe. So what was the difference in the method? So you can actually understand and make sense of this, but only if you have the methods alongside the data. And that's my constant refrain is data sharing should really be going hand in hand with method sharing. We've made enormous strides. So Emily mentioned my time on different journals, both Journal of Cell Biology and PLOS Biology. In both of those locations, I worked a lot on policies around sharing data, whether it was microscopy image data or whether it was just all of the data relevant to reproduce information in papers latterly at PLOS Biology. We do really well now at sharing data in different repositories here, there, and everywhere. So tons and tons of data shared out there. However, the method side of that is really lacking still now. So there's a brilliant quote from an evolutionary biologist there. He said that nothing in biology makes sense except in the light of evolution. And you can see my variant of that quote, which is nothing in research data makes sense except in the light of how it was generated. And that is where protocols.io comes in. So it is an online tool to share method details. And our mission's pretty simple and want to make it easy to share method details before, during, and after publication. So anyone can come along and make a free account on protocols.io. If you do, you have most functionality available to you, but you have a limit to the number of private files that you can have. So the business model is selling, it's very much like GitHub, which is providing access to having more private space if you want to generate and develop more content privately. But you can do everything, and most importantly, you can publish and make your methods open access by publishing if you want to do so, and that can all be done for free. Anything that's put on there and published will have a DOI and will be available with the CC BY license. So it's very much been mission-based around open methods. Business model underlying is just because sustainability does need to happen at some point in time. So here it is now. This on the left is a screenshot of a good page to come and play around on if you haven't looked at it before, which is just protocols.io/welcome. We were acquired by Springer Nature right about a year ago now. It was in July last year. So are now still striving to be independently sustainable within Springer Nature, but are much better supported as a small team, as a part of a huge organization at this point in time. But apart from them now being kind of our owners, the business model and the way that we function as a tool has not changed at all, really. So still has the open access repository, can be used across all research disciplines. It's collaborative, so it works like, if you're familiar with a Google Drive or a Teamspace, you can have a space where you invite members, they can all access the content in that space and work on it and collaboratively edit and develop and make changes in there. And we're very careful, obviously, about security and archiving and mirroring content. It really does tick all of the FAIR acronym checklist elements, though. So as a repository, it works interoperably with the DOI as the persistent identifier. It's something that is findable, accessible, and we've got lovely reuse functionality, actually. And then there's just some stats to show you that there is a lot of users, a lot of public content, and a lot of people using it, that private side, to develop and optimize and actually record run records of the protocols on the platform. So this just shows how it can bed right into the workflow. So if you do have access to using it for kind of private file development, it can be used throughout the entire workflow. So you can start a project and look for methods that maybe already exist on the platform, or bring in content from outside, methods that you know you'll be using, and you'll start to plan and outline, put those methods on, identify protocols, add information about equipment, materials, sourcing information, and can start to share and collaboratively edit with colleagues. You can get feedback via comments. There's some lovely dynamic functionality to comment on the protocols, so you can do all of that on there. So I'm moving around one of the Teams windows as I talk so I can see where things are. And then you can actually start performing the methods. So when you have any method on the platform, you can make a run of that and record information in that run, where you can actually gather observations, make comments, and put information in. And then obviously you'll be assessing the results of that protocol run, if this is a kind of experimental approach, and was it successful. If it wasn't, and you need to tweak, make some changes, or you're trying out variables to optimize, you might end up looping here a little while you actually get that to work as well as you possibly can, get the results that you need. And every time you run it, you can create that run record, which will be date and time stamped. So you'll have information about what you did, which you can correlate with the data that you generated on a given day. So you'll have a really nice record of everything you've done. Hopefully, eventually, you succeed and have successful protocols that are working for you. And at that point, if you're writing up a research paper, you can post the protocols publicly on Get a DOI on protocols.io, which you can drop into the materials and methods of the related paper. So you can link out and say the paper can just have minimal methods info about what you did, and then say comprehensive information is available on protocols.io, and have that link to the protocol itself. So I've got a screenshot. This is the last slide, and then I will go really into showing you the platform. Keep an eye on time. I don't want to take up too much of the time. So this is a very well used protocol on protocols.io. You can see it has its title. You can version protocols. So if you've posted one and published it, you can come back and publish subsequent versions, and they'll all be linked to one another. You can compare them back to one another to see what changed. If you have included the DOI in a paper, we can capture the reference to the paper here at the top of the protocol, and then obviously the protocol's own digital object identifier sits there as well. Have the authors, and then you have an owner on protocols.io, who's the person that's actually kind of owns that file, if you will, and will be alerted if comments are added. This is an extreme example in that it has over 1,400 comments on it. We see this as really, really constructive. Back and forth happens on protocols.io where people ask questions about methods because they're trying to reproduce, and I'll show how that looks in just a moment. But when someone does come along and replicate the method and get it to work, they can click, we have call it the works for me button. And on this one, 61 other researchers have managed to replicate, get this protocol to work, and have reused it. So we can capture that information here. Other users can also make a fork of a protocol. So that's GitHub terminology, really, which is sort of what protocols.io is originally modeled on, in that someone else's protocol is available publicly. I can fork a copy of it for myself, which will be private, but which I can change if I've got slightly different equipment, or I'm applying to a different chemical if I'm assessing toxicology, or a different cell type, for example. I can make a variation of that protocol in which I adapt and make those changes to make it relevant in that way. There's that run button. So if you want to run it, that's really easy as well. You scroll down the page a little bit. So obviously you saw that had abstract, and then it goes into a step-by-step format. Authors can really easily create sections, give little subheads, and that automatically generates a table of contents as well that can be used for navigation. And that's what the comments look like. So at an individual step, I could click, add a comment and ask a question, and the owners or author of the protocol will be alerted and they can reply. And that's what I mean about this really constructive feedback element to the platform, which is really, really good when there are these methods that other people do very much want to develop and make use of in their own labs. I can hear the binging going of people joining. So there's lots there people can do, can compare their versions, and you can publish, submit for peer review, etc., etc. Let's see. So I'm going to jump out, and these are the things that I'll just try and demo quickly, actually, in the platform. If I can jump around in here. The first one, I won't really show this, but I think Paul might be going to show this. This is one of his. You can see he has lots of nice functionality in one of the protocols that he's added before, and that's on a publicly available one. But the behind scenes look, here is a workspace. So this is like the Drive location where I could invite whoever I wanted to be a member in this workspace, and they could work on the same content as me in there. You can create folders for structure. You can set it up however works best for you, and make it work, either folders per project, folders per protocol type, or here, depending whether they're in development or they're published or working, etc. All of these different elements. And here it's very easy. We could make a new protocol. You can also make a collection. So if you have some kind of higher level overview of multiple protocols, you can put them all together in a collection. Or if you have multiple that will be a part of one published research paper, you can link to all of the protocols in a collection that way as well. So, and when you're in here and you've clicked on one, you can see you have a lot of options on the right-hand side. All of that functionality is also available in the editor. And we've got British people on in a Blue Peter fashion. Here's one I prepared earlier. So here is a very basic demo protocol that I have created. When you first start out, it would be completely blank, but you would see this new, which is where you can very easily add a new step or add a new section. So if we made that section 6, I need to delete some of these. I've done it a few times in demos. You can see that that just creates you this new color coded section, which is always reflected in that table of contents, which is what I alluded to before. What we also have over on the right are these things, we call them components, and they are where you can capture variables of different things that might be useful in the context of your protocol. So if you are adding volumes of something, for example, here we've got 50 ml reagent A, you can change the units, add the contents and quantities, etc., like this. If you've got a lot of different reagents, you can specify specific reagent information. We pull in a lot from catalog IDs, from vendors, but also some users. So this is a user at Sanger who's added this one. We'll add them in. You can create your own. That way you can be very specific about catalog ID, exactly what the reagents are, which is obviously very helpful for reproducibility. Timers, so if you have durations, you can put that. Temperatures. This next one down is what we call a smart component. And I know Paul has used this, and we are still building out functionalities to improve this, but you can define key value pairs in these where you put in what it is that you want the observation to be, and then you either require or make it not required that an answer be added during a protocol run. So you prompt people. So this one, excuse me, this one requires an answer to both of these questions, but I could change what the text is. So if it was, count of cells or percent dying out or something like this, you can add in whatever the relevant thing is as your smart component. We can input tables, and you can include basic Excel formulas too. So if you actually observe and put in numbers during a run, the calculations will happen on the fly. So this is obviously a very trivial sum example, but I'll show you how that works through the run in just a second. Can include videos and images. So if it's a tricky procedure or you want to show exactly setup, you can very easily add those from a mobile phone or device, put them in directly. If it's a larger one, you can embed something that you've already uploaded onto YouTube or Vimeo with embed code. So if I edit that one, that's how that's been pulled in, is embed code from the share link of a YouTube video. So it's very, very easy to put those in. I will quickly show you the run functionality. Always want to give us 5 stars, but I figure I probably shouldn't do that. So I mentioned the run. We will click to run and just start running the protocol. And this allows me to step through every step of that protocol, but actually there's some other nice functionality. If I've used the reagent volume components that I was talking about previously, I actually can scale this if I want to. So if there's a master mix to be created, but I actually want to make more volume today, I can do that very easily by scaling up, and it will reflect what I did, what change I made in that note here. If I've used the timer, I can run the timer as I dynamically step through. And then I'll say that step's done. It's not going to let me go past this one until I input something here and here, and then it will let me move forward. So you can require these observations to happen as you go through. If I say I want to edit this step, this is where I might be putting in some observations of something I'm actually observing during the protocol. And you can see that the sum happens on the fly. And again, I can save that. And when I eventually save out that run, all of this information will be included in that run record. So if I put that in my run records, and we'll just put demo EBTC at the top and save. If I don't click that date time to say that it's finished completely, because I only got partway through, I can come back and finish it later. So if I go back to that file manager but now I go into the run records. It's here. It's a run record. If I click on it, actually I can resume. So I can pick that back up and carry on. So if it's something that happens over multiple days, I can leave it and finish it tomorrow. Or another member of my workspace could actually finish it if they're in there as a member of that workspace. So if it's something where researcher A is going to do the first part, researcher B does the second part, you can actually hand that over and the run record will reflect who did what as well. So if we go back into that run record, we can say, oops, where did it get to? Finish, and we're not going to completely go through it, but we'll say finish run. And so that's finished out. And if I go back now and refresh, that will be a completed run record. So it's turned green, it's completed. And if I open it out, what you can see is we've got the record of the things that have been added, the data that was put in. And if I look under, the history, you can see who the person is that made those steps through and anything that they changed or did. If I'd added comments, I'd be able to see those here too. I know that Paul's efforts, they then export these run records as JSON with some computational add-ons so that they can extract information. So you do kind of have to be a super user to go to take it to that next step. But if not, within the lab, you at least have this record that you can keep of a protocol like this. Let's just jump across and show you a couple of other example things. So this is that screenshot that I showed earlier of the protocol with all of the comments on it and the versions. So we can see all of those earlier versions and the dates. If we clicked over to the right, we could compare any version to another version this way. There's that link across to the eLife paper. This one has quite a complicated lot of different sections, so we can navigate down using that and maybe find, no, I can't find any comments, somewhere where there have been comments added. Okay, so now there's quite a lot. So here's where someone has asked a question and the owners have answered the question, and I could put another question in if I wanted on that step and the authors would be alerted to that. They are automatically publicly visible unless you select private, but most people clearly put them on publicly because then that turns this into a real public FAQ, which is just a super extra information availability. If you ask the question, everyone else will be able to see the answer. But actually, if you come along and you think you have a question, maybe it's already been asked and answered. And we have nice metrics. So that reuse to see how protocols are being used by others, if you've made them available, you can see the number of views. Again, these are cherry-picked big examples. This one's had lots of views, bookmarks, forks, comments. You can see where they've been cited as well. And the very last one, I'll show you an example, and I promise then I'll let you talk, Paul. This one is just to highlight what the forking can do for you. So this was coronavirus sequencing protocol, version 1, published January 2020, very early on, before most people knew anything was happening. And the reason they got it up so quickly is because they could fork it from a different virus sequencing protocol, an Ebola virus sequencing protocol. And this just shows you can compare to the fork as well, and how the comparison looks like if you do that ever between either versions or a fork to what it was forked from. And in this one, there's just different volumes of template RNA very early on, different notes, and then different primers, obviously, for these runs that are happening. So lots of nice functionality. And you can also visualize forks. So if lots of people do reuse your protocol, again, it's a very extreme example. Many people did come and make use of this coronavirus sequencing protocol. We can actually visualize all of that reuse, which is just kind of lovely example of open research, really open science, and how sharing methods can be very, very useful globally, especially in public health situations. But this does happen to a lesser degree on other protocols. This is exceedingly extreme, but it just shows you that actually we can really capture with that fair perspective all of the reuse of the protocols that we have. And finally, I'll just show systematic reviews. It's it's not the most obvious fit, but we can accommodate them. This is just one published one. And people tend to kind of just add the sections and things into steps on protocols.io, and Paul will talk more about case study that he did with the platform. But I did want to note we do have some peer review options if you're in a protocol and looking to publish it. Sorry, it's all gone slow finally, of course, just as I want to show you something at the end. If you're looking to publish it, we have the options of publishing to get a DOI are very obvious where they are, but we also have peer review options. At the moment, we have 3 journals on here. Very soon, we will have a partnership with Systematic Reviews, the Springer Nature Journal. So that will be getting added as well. And we're really hoping to have some templates available that we'll be working on probably in the next 6 to 12 months, so that we can create more of a template structure for things like systematic reviews, where authors can come along and complete that information that's been nudged and made available as a starting point. And I will stop there and let Paul let you take over. Sorry if I spoke too long.
No, it's great. Thanks very much. Much better that you speak too long than I do, I can assure you. Right, so so what I'm going to do here is actually first thing I'm going to do is share my screen. That'll probably be helpful. So let me just grab this window here. There we are. I'll just pop that back up in size again. So Emily, can you just confirm you can see a browser window there with protocols.io methods templates showing?
Yes, I can see it. It doesn't appear to be in presenter mode, but
it's not supposed to be. I'm going to be jumping around all over the place here, so making everyone feel dizzy. So I just left it in regular mode. Also, I should just draw everyone's attention to the Q&A function. So if you have questions that you want to type in, then please do. We'll also, everyone should have their mics enabled so they can unmute themselves. If you want to just ask a question verbally, then also feel free to unmute your mic, turn your webcam on. If you just use the raise hand function, just so we can create a stack of people, if we've got several people wanting to ask questions, that would be fantastic. So as Emma said, like EBTC did use protocols.io for a case study to see if we could use the system for helping people report systematic review protocols in a way that complies with best practice conduct standards, so conduct guidance, and also with reporting checklists to kind of take the pain out of having to figure out how to make your own systematic review checklist compliant. If we could provide a template for doing a systematic review where we took care of all the checklist and standards compliance issues for the user, and the user could just then plan their own systematic review in with confidence that they would be standards compliant when they used that. Probably could have said that better. Anyway, it's all described in a manuscript here, and I'll share the links in a bit. It's quite a long paper, so obviously I'm not going to go through everything there, but it does kind of go through everything that we were trying to, all the value trying to squeeze out of protocols.io. The reason that we did a systematic review template was that systematic reviews happens to be the thing that I know best. So when I'm working through this, for everyone here who does kind of in vitro research, like bench toxicology and things, just think about how what I'm showing you as we apply to systematic review could be interpreted for things like OECD test guidelines or your own experimental methods that you could turn into a template that someone else could then use, basically. Also should emphasize that this technology is pretty new, so we're learning how to use it at the same time as you are. So if you have any thoughts and comments and points for discussion, then that would obviously be very welcome, because there's a lot of things to square off here. It's a really interesting space. So I'm just going to quickly jump over to the protocol in question. So this is this systematic review protocol planning template that we created in protocols.io. It has, as you can see on the left here, 1, 2, 3, 4, 5, 6, 7, 8, 9 sections. It's quite long, and there are 44 steps in the whole process. Probably sounds quite intimidating, but the nice thing about protocols.io is that when you start a run record, it just presents you each step one at a time. So I hope this is working. So the first step of the systematic review protocol that we have basically asks you to create an open science framework record for the project, because you need somewhere to put your data when you're doing research. So we give a little bit of guidance here on how to complete this step. And then there's a completion point. So once you've created your OSF record URL, so that's Open Science Framework, it's one place to put research data. You would write the URL in here, so url.com. You'd enter the title of your protocol. This is Paul's protocol. And then you've completed the information that is required, and then you would just click done. And that's the first step of your systematic review done. You found somewhere to put your data, you've created a bucket for your data, jolly good. So that takes us to the second step, where an important part of doing a systematic review, of course, is it's a complex multidisciplinary project. So you want to make sure you've got team members who can cover off each of the core competencies of systematic review. These are best practices as described in the Costa standard for conduct of systematic reviews in environmental health, and then the Prisma P reporting checklist, item 3B. So what we did when we were creating this protocol template was we looked at 6 or 7 existing standards and checklists, abstracted from those all of the implied steps that you'd have to do in a detailed planning process, and then cross-mapped each of the steps of the planning process with the steps and the recommendations and the checklist items in each of these reporting standards or conduct guidance. So you, when you use this template, can see which parts of those checklists you're ticking off as you go through it. So you'd go through this and you'd enter in who's your information scientist. That would be someone like me. Someone like Emily would be our evidence appraisal methods person. We'd have a statistician in, so my friend Steve. And you'd go through, you check all these off. Then you've completed the competency step. You know everyone in your team is going to be doing each part systematic review, and then you're done again. And that takes you to step 3, which is about identifying information management practices and tools for each stage of the review. So then again, we've got the crosswalk here to Costa guidance item 1.1.2. We've got Prisma P, that's checklist item 11A that talks about the information management practices, and then Prospero, which is somewhere where you can register systematic review protocols. They have some information that they want you to provide when you upload your protocol, and that's item 26 from the Prospero reporting checklist. And you just state the software that you want to use. And we know when we do systematic reviews that you need a reference manager, a knowledge management tool. You need to know what your systematic review software is, what your statistics software and packages are, and what AI support tools you're using are. So you go through, you complete all of those, you would hit done, and you would move on. And it just goes on and on like this through your 45 steps. And when you finish it, you end your run, and then you've got data that you've collected at each stage of the planning process, all kind of nice and tidily organized for you, which if you're crafty, you can export as a JSON file. And then you can use some code to scrape out the data from the JSON export and automatically assemble that into a draft manuscript where you know you've done all the steps you need to do to be standards compliant. You've got all the information you need to complete the reporting checklist. It's all already in there. So you know you've got a complete documentation of a complete systematic review planning process. And hopefully that ends up being very useful to people because it means they don't have to think about these things themselves when they're already in the nitty-gritty of trying to be topic experts and actually doing systematic review themselves. So that basically covers that off. We are also giving people an opportunity to publish these types of protocol templates. They don't have to be done in protocols.io. Emma showed you've got 3 publishing peer review options. We've actually just launched a special issue for exactly these types of things. So you can submit these to us as well. And there's actually a prize to go for the best study methods template that's published in our journal before the end of the current calendar year. So if you have any interest or questions about that, then do please let me know. And otherwise, I think I would just at this point like to get onto questions as quickly as possible. So thank you very much for listening. I'll drop all these links into the chat and questions as well. So if anyone has any questions or thoughts, that would be really, really welcome.